Hi, my name is Karp and I recently finished my 2-month placement at Douglas Henley Moir in Macquarie Park. Douglas Henley Moir is a private lab which performs high-quality diagnostic pathology tests for clinicians and healthcare professionals, private hospitals and nursing homes. During my placement, I was able to work in 3 different departments: prenatal testing, microbiology and molecular pathology. Prenatal testing involves screening the overall health of fetuses in expectant mothers by measuring protein levels such as AFP, alpha-fetoprotein, that the fetus secretes into the placental membrane. AFP should exist within certain concentration at different stages of development. By taking a placental sample, AFP concentration can be measured by an immunoassay. This involves binding the AFP to a surface of antibodies, washing away the waste, and then binding another antibody tagged with a fluorescent marker, almost like an antibody sandwich. The amount of fluorescence which can be analysed is proportional to the amount of protein and can therefore screen for the overall health of the fetus at different developmental stages. In microbiology, the same species of bacteria can be resistant or susceptible to different antibiotics. Sensitivity testing was used to see which antibiotic could be used to treat which bacterial infection. Bacteria is spread on agar plates so that it can grow freely. However, multiple small discs that release different antibiotics permeate through the agar. If the bacteria is susceptible to the antibiotic, it will leave a clear zone where it can't grow, or it will grow freely up to the disc if the bacteria are resistant to the antibiotic. I also worked with PCR to detect Giardia and Cryptosporidium. The basis of PCR involves amplifying specific regions of DNA unique to the pathogen to detect if it's present. If there is no amplification, the species is not the cause of the illness. First, lysing the cells, the DNA is split into single strands with high heat. Then, with a lower heat, free primers and nucleotides are bound to regions of DNA wanted for amplifying. The DNA primers and nucleotides create a double-stranded region of DNA, and the process is repeated. The nucleotides are often tagged with fluorescence, which is proportional to the amount of DNA present, and due to the exponential growth, there is a lot after the 40th cycle. Some of the ethical dilemmas involved in patient care can be really daunting. As an intern, we are faced with learning lots of new things at once while being expected to maintain a high level of accuracy. Something as simple as labelling the samples wrong can mean that the patients don't get the correct results, and their health will suffer as a consequence due to a treatment plan that might be for someone else. As mentioned before, the prenatal testing department is a screen, which is done on the fetus via the mother's placental sample. This may indicate an increased risk of developing Down syndrome or another abnormality. If something is wrong with the baby, mothers nowadays don't necessarily want to terminate the pregnancy, but they want to be informed so that when the baby does arrive, it can be ready. If there is a mistake with the sample, such as labelling, or the sample was incorrectly prepped, the mother may not be able to make an educated decision on her newborn baby's life. So not only are there ethical ramifications so that the mother can have a choice in the pregnancy, but the legal ramifications for the lab being liable for a life that wasn't wanted by the mother are serious and possibly potentially damaging for all parties involved. Overall, in the lab, I had a very positive experience. My role was to learn as much as possible during this placement. Unfortunately, however, for a lot of the work you need to be specially trained. But I was able to learn very quickly and collaborate with workers to complete the task more efficiently. This involves small but still very pivotal roles such as setting up extra plates for further species identification, preparing Vitex sensitivities, or even preparing samples for tuberculosis screenings and more. The emotional intelligence I developed seeing which staff members needed the most help was pivotal to my success in the lab. However, at the start it was still very difficult to gauge, but through observing staff's emotions and the overall structure of the lab, I was able to contribute positively to the lab's time management by seeing who needed help when. From my placement, I can also see how accuracy and attention to detail not only make their work high quality, but allows the patients behind the samples to get a very quick and potentially life-changing results. As a worker, I can definitely see improvement in these areas for myself, where I'm now double-checking my work more thoroughly and taking more care when performing tasks the first time around. I noticed throughout my placement that pathology workers are limited in their training. What I mean by that is that they know everything they're meant to do for their jobs, but they don't necessarily have to use their brains in new ways when they have all the information they need. The department managers, often pathologists or PhD researchers, were the people who made the department viable by creating innovative solutions to, at times, problems that we get unknown. For example, one of my supervisors created the prenatal test now used in the department, and the other set up the National Bowel Cancer Screening Test. This makes me think a career in pathology is not for me, but maybe in research or even as a doctor having a closer relationship with the patients. I really like to thank everyone for the opportunity to be in the internship. I had a wonderful time, and at least for the near future, pathology is definitely part of my career as I was offered a job at the end of my internship. Thank you.